In this video, we're going to look at the process of transcription, which is the production of RNA from the DNA template. It's important that you understand the difference between transcription and translation, as getting these two mixed up can often lead to a lot of confusion. In simple terms, the process of transcription is taking the correct DNA sequence, splitting it into two, and then making a single-stranded RNA copy of the one DNA strand. However, as usual with most of these things, something that starts off sounding simple can be incredibly complex. The first thing to remember about the process is that the resulting RNA molecule is not an exact copy of the DNA. Plus, as you know, RNA uses ribose and DNA uses deoxyribose. The reason that the RNA is not an exact copy is because RNA uses uracil instead of thymine. This means that when we get an adenine in the DNA sequence, then in the RNA sequence, instead of matching it with a thymine, it will be matched with a uracil. Now, here I need to introduce a new way for how we refer to DNA sequences, and that is sense and antisense strands. And these terms refer to the sequence that gives, when decoded, the protein sequence. Anyway, the sense strand of DNA is the strand that contains the information that we decode to make the protein, whereas the antisense strand is the complementary, the opposite strand of DNA. When we write out a DNA sequence, we write it with the 5' end on the left and the 3' end on the right. But we also, if we are only showing one strand of DNA, write out the sense strand. And if we're showing both strands, then we write the sense strand on the top and the antisense strand underneath. Why do we do it this way? Well, it's just the way that we do it. To add a little more confusion to this, we sometimes refer to the sense strand as the forward strand and the antisense strand as the reverse strand. And finally, I must just mention streams. You will often hear terms like upstream and downstream used when people are talking about DNA or RNA. All that upstream means is in the direction of the 5' end from the point we're talking about, and downstream means towards the 3' end from the place we're talking about. Our hereditary units are genes. It is these genes or transcription units that give us our proteins via messenger RNA. Or they may produce RNA molecules such as transfer RNAs, ribosomal RNAs, and microRNAs, where the RNA itself is the functional end product. And don't forget, all of this is done in humans using only 20,000 genes, which is about 1% of our total genome, with a large chunk of the remaining DNA, some 80%, producing non-coding RNAs, which have a role in regulation. RNA polymerase is responsible for making RNA using DNA as the template. In prokaryotes, there is just one RNA polymerase, but in eukaryotes, there are three, which are cunningly called RNA polymerase 1, 2, and 3. And each polymerase has a different role in the cell. For example, RNase polymerase 2 makes all the messenger RNA that encodes for proteins, plus some microRNAs, whereas RNase polymerase 1 and 3 produce the other types of RNA used in the cell. The process of messenger RNA production can be split into 5 steps: initiation, production, 5' capping, splicing, and 3' polyadenylation. In step 1, initiation, activators bind upstream, often thousands of bases upstream of the gene. These activators then help assemble the required proteins: the mediator, chromatin remodeling complex, the RNA polymerase, and transcription factors at the tartar box, which is closer to the gene to be transcribed. The tartar box, along with some other sequences, is what is known as a core promoter, and it's located just upstream of the gene, and it is in this region that the transcription machinery builds. Upstream of the core promoter region, you will find the regulatory promoter region, which, as the name suggests, regulates the transcription of the gene. Now, the tartar box, which is a core promoter and is located 25 to 35 base pairs upstream of the transcription start site, consists of the sequence TATAAA, and is a key recognition site for the binding of the transcription machinery. Next, in step 2, the DNA is transcribed into pre-messenger RNA molecule at a rate of 1,000 to 4,000 bases per minute. The next steps, 3, 4 and 5, require a number of enzymes to form specific tasks and to process the pre-messenger RNA. These enzymes are carried on the C-terminal of the RNA polymerase on a series of phosphorylated tandem repeats, and as the pre-messenger RNA emerges from the polymerase, these proteins bind to the growing RNA chain and carry out the required modifications. In step 3, the 5' end of the pre-messenger RNA is capped with some modified nucleotides, and this helps protect the molecule from degradation by the cell. Now, before we go any further, we need to have a quick reminder about genes. Genes in eukaryotes consist of exons, the bits of DNA that code for expressed proteins, and introns, the DNA between the exons. So this means that an RNA molecule will contain exons and intron sequences. So in step 4 of the process, the introns are spliced out of the pre-messenger RNA to leave just the exons. And this process is carried out by a large protein RNA complex called the spliceosome. And this process is very interesting as it can make different messenger RNAs from one gene, and hence the cell can make different proteins from one gene. Well, the spliceosome can process the pre-messenger RNA and use exon skipping to exclude certain exons from the mature messenger RNA molecule, and hence produce different versions of the protein. So this means that we can produce more than 20,000 proteins, as the cell can use exon skipping to make more than one type of protein from a gene. An example of this is alpha-tropomyosin gene that can make at least 5 different versions of a protein by missing out different exons. The final step, step 5, is the addition of a polyadenylated tail to the 3' end of the pre-messenger RNA to give the final mature messenger RNA molecule. The polyadenylated or poly-A tail is just a long tail of adenine bases that, like the cap on the 5' end, helps protect the RNA molecule from being degraded. Now, steps 1 to 5 all happen in the nucleus. And once we've got the mature messenger RNA molecule, we need to export it from the nucleus to the cytoplasm where it can be translated.